presentation. Our speaker today is Dr. Will Redfern. He's currently the Vice President of Quantitative Systems, Toxicology, and Safety at Certara, where he leads a team that applies computational approaches to assess the safety of pharmaceuticals and other chemicals. Will is an experienced safety pharmacologist, having worked at Syntex, Quintiles, and AstraZeneca, and he was the former president of the Safety Pharmacology Society. Today's presentation is Introduction to ToxStudio, Smart in Silico Tools to Aid Translation from your in vitro to in vivo studies. Will, welcome to today's event. I'll now turn it over to him to begin the presentation.
Thanks, Suzanne, and good morning, good afternoon, and good evening, wherever you are. Thank you very much for dialing in and taking time out of your busy day. So first of all, to say that we have a virtual booth at the SPS, which is actually still open. I think they're open for 12 months, so they're there for any time that suits you to pop in and click on any of the assets that are on the back wall of the booth and leave any questions, and hopefully we can address them. So quickly, just to reflect on Certara, who we are and what we do. So this is really that Certara optimizes R&D productivity, commercial value, and patient outcomes through its unique portfolio of model-informed drug development, regulatory science, and market access solutions. And there's an interesting statistic that over 90% of all novel drugs approved by the FDA in the past 6 years were supported by Certara software or services. And this is a selection of some of our software products along the bottom, and you may have encountered or heard of or used some of these already. So I'm going to talk to you about the QSTS tools that are in development. So first of all, to define QSTS, this is a quantitative or computational framework to support translational drug discovery and development by integrating knowledge on biochemical, biological, physiological, pharmacological, toxicological, and clinical systems with a focus on drug toxicity, safety, and toleration. And this is our team, and we're a team of modelers and pharmacologists working together. In terms of our publications, the blue stacks show the increase in the number of publications using SimSip, which is mainly a DMPK, a clinical targeted engine. And the tips in red show publications that are QSTS related. So the team's been making its presence felt in recent years. And we're also backed up by some excellent software development guys who've also worked on the SimSip simulator. So I'm not going to read out the background and CVs to everyone, but just to sort of summarize who's a modeler and who's a pharmacologist, although the edges are blurred sometimes. So Mark and myself are the two experienced pharmacologists joined by two new graduates, Gus and Katie, and the team, Kieran, Jeremy, Sebastian, Mitras, Vipul, Emily, and Zofia of modelers. So the team's activities are divided between activities on the QSTS consortium, which we set up, and our first pharma partner is Amgen. And the next bubble is the products developing for ToxStudio. The grant work, 3 EU-funded grants in there, and consultancy work. So really it's to say we'd like you to join us in the consortium. The products are going to be the main focus of today's talk, one in particular. In the grants, we're already working with several of you. And in terms of consultancy, if you've got any challenges in terms of relatively short projects, please come and speak to us. So some of you who know me well might be thinking, well, you know, you're known for your in vivo work. Why have you suddenly moved to this newfangled in silico stuff? And I'd make the point that it's not been that sudden a jump. This review article dates back to 2002, and one of our concluding remarks under future trends was that we are still some way off an in silico model of how a drug's interaction with a particular receptor or collection of receptors located across various physiological systems, also taking into account pharmacokinetics and repeat dosing, will affect entire organisms. Well, we may have been some way off this in 2002, but hopefully I can show you that we're getting closer to it now. Also, if we fast forward a bit to 2014, this was a talk I gave at the SPS where I was really talking about the transition to in silico approaches. And so, you know, this has been something I've been thinking about for quite some time. I also was going back on this slide deck and found this towards the end of the presentation. So my crystal ball was working well back then about predicting a virtual meeting. It's just a shame I don't have so much luck with picking lottery numbers. And as for in silico being newfangled, well, you can trace systems modeling back probably to the 1950s, and Dennis Noble, who at the time was a PhD student at University College London, subsequently moved to Oxford. This is a publication from 1962, and he had an earlier publication in Nature. And what he was doing was modeling the cardiac action potential from the Hodgkin-Huxley equations. And these are computed action potentials. And the computer that he used was this university computer, Mercury, which he had to cycle in to program it in the dead of night when no one else was using it. So this was really sort of heroic stuff, but this was probably, he didn't feel it was ahead of its time, but when we look back on it now, it certainly was a very early pioneering work on systems physiology. So it's not at all newfangled, it's been around for several decades. So the various tools available for in silico safety pharmacology, if we look at them starting on the left, there is QSAR for undesirable off targets, and this has been around quite a while. Then you've got omics, bioinformatics, and also use of soluble biomarkers. So these are sort of informatics types approaches. There's natural language processing, best component of that that's best known as text mining. Safety databases, and you saw me refer to one earlier from Satara D360. Safety data visualization tools where you can overlay multiple data types, and then the effectively the modeling and simulation, including quantitative systems, pharmacology and toxicology models, PK/PD, and PBPK. In amongst this sits machine learning, which can talk to several of these. So there's quite a lot to think about in the in silico world, but in terms of our team, we're focusing on what we have a background in, and that's in the modeling and simulation, and also in the output of safety data visualization. So the concept of ToxStudio is that it's a core interface in terms of knowledge and modeling integration. So it will take in vitro data analysis and import modules, and we've also tacked on here our SIVA toolkit. So that's the in vitro data analysis toolkit. We'll also talk to modeling modules, including your own if you've got any built. It will talk to SimSip, which a large number of pharmaceutical companies use, or they actually have SimSip for their clinical DMPK predictions. It will also receive other data types, talk to SimSip Animal, which is an animal version of the human SimSip, and then provide visualization data outputs. So the problem we're trying to address is a familiar one to this audience, which is the stubborn problem of safety-related attrition. And these different separate analyses date back to the 1960s, and as a percentage contribution to the overall attrition of drug projects, it's been sticking around the 20 to 25% of the total. This is being addressed by large pharma in various ways, starting with anticipation of target-related toxicity by target safety profiling. Then when you've got compounds, early de-risking, avoiding molecular structures and phys-chem properties associated with known toxicities. Hazard detection on the compounds from in vitro screens and assays, and from the use of omics and biomarkers in vivo. And then better risk assessment, where you synthesize all the available in vitro and in vivo data and juxtapose it with the clinical tissue exposures, or the predicted ones. So while this is beginning to have a positive impact in some companies, the overall picture across the industry is unclear. And in any event, there's always room for improvement and no room for complacency. Although there does appear to be some complacency, because this was a live online survey run during the virtual SPS a couple of weeks ago. And in this survey, in terms of their response to this question, 42% of participants considered that their current preclinical assays and models are adequate to reduce attrition. So the question was, do current preclinical drug discovery and safety assessment strategies, which rely on primary assays including cellular, tissue, organ, and in vivo systems, adequately reduce efficacy and safety-related attrition? 42% were of the opinion that they did. So if we also look at regulatory influences, and we know that within safety pharmacology, regulatory drivers are very important. Within the field of PBPK modeling, there are two recent guidelines from 2018, one from the European Medicines Agency and the other from the FDA. And there was also a recent FDA review on the applications of PBPK modeling in clinical development. Most of the applications were drug-drug interactions, so this is where you use the modeling instead of doing a very expensive clinical study. There was also a good chunk on pediatrics, so again, instead of doing clinical studies in children, you can predict from other data. And so on. And then there's a small chunk called other, and we feel that safety currently resides in there, and we want to expand that. And of course, within the safety pharmacology arena, there's SIPA, and the particular aspect of the 4 columns of SIPA is the in silico reconstruction of human ventricular cellular electrophysiology. So in other words, the FDA have embraced systems modeling of the cardiac action potential. And there's a nice review on this here. If we look at regulatory trends in the non-pharmaceutical sector for chemical products, they're moving away from in vivo testing. So the EU banned testing on animals for cosmetics from 2009 after signposting it for several years in advance, and then they extended it to a ban on products imported into the EU. And in the chemicals and agrochemicals sector last year, about a year ago, the Environmental Protection Agency, EPA, in the US proposed a ban on testing in mammals from 2035. So there will be some milestones leading up to that. Now, clearly, these two bans do not affect the pharmaceutical industry, but we need to take heed of the direction of travel. Another aspect that we should consider is that as at any time, really, a large swathe of highly experienced safety pharmacologists and toxicologists are retiring or approaching retirement age. Nothing new about that. In previous years, this would not be a problem, as they would gradually and progressively pass on their knowledge to a new generation of safety scientists. But there have been some dramatic changes in recent years. First of all, there's been a shift to outsourcing of in vivo GLP studies and a lot of in vivo studies per se, obviating the need to hire experienced in vivo safety pharmacologists into pharmaceutical companies. And secondly, trimming down to a lean organization, often leading to key person dependency. So if somebody leaves the organization, it may be difficult to recruit someone with the same level of experience. So we thought if we could bake in some of this holistic in vitro, in vivo findings. I think my slide set's just slipped out of slideshow mode. Apologies for that. If we could bake this into an in silico tool, this would assist a younger generation of safety scientists make sense of what may be unfamiliar in vitro and in vivo data in order to make the right safety calls to project teams. I'm dropping out the slideshow mode, so apologies for that.
I think you're fine now, Will. Yeah, so it dropped out, but yeah, now you're back.
Yeah, good. So if we draw the current preclinical clinical safety paradigm in this this slide, it's showing a flow from the top is the start of drug discovery down to clinical development. So this is where you have project compounds that are synthesized, and then you will, from safety perspective, run them through traditional 2D in vitro safety assays, and then move on now to more sophisticated 3D microphysiological systems, multi-electrode array type complex assays. And this informs the chemistry. And also, you'll run secondary pharmacology profiling, which also informs the chemistry. And some of this will trigger further 2D in vitro or more complex in vitro assays. You'll then move on to in vivo preclinical safety testing, which will take on board animal ADME or DMPK data, and then on to human. So where we see the involvement of in silico tools that we've got and are developing are to basically turbocharge this by supplementing these assays with in silico tools, and this will improve the translation to the next stage and on to human safety. So this shows where our various tools fit into this flow chart, and I'll return to this later. So the first area we're going to address is secondary pharmacology. Why is this? Well, you're largely stuck with the safety liability as the primary target. There's some room for maneuver. Whereas if it's an off-target activity that's causing a safety issue, you can either deselect a compound from a set of compounds, or you can dial out or dial down that off-target activity by changing the chemistry early on. So most pharmaceutical companies screen their compounds of interest against a broad panel of off-target receptors, and they may spend typically $500,000 to $1 million doing this each year. And we'd ask the question, is the best use made of the data? Certainly the regulatory agencies ask questions on the profiling data, And there's been a growing prominence of this discipline over the last decade, with some nice reviews coming out from the pharmaceutical industry, including some multi-company reviews, and including this consensus paper from Joe Bowes et al. And also some reviews from the FDA as well. So it's clearly a discipline that is of great importance within safety pharmacology. And one of these reviews was actually based on a survey where they asked 18 companies various questions. I've picked out 3 questions here. So the first was, are your pharmacological profiling data included in regulatory submissions? And the vast majority said yes. Are the data put into context of the in vivo non-clinical results? And again, the vast majority answered yes. The third one, did you get regulatory questions on the significance of the in vitro secondary pharmacology results? There was a bit of a 50-50 split, but I certainly recall from my AstraZeneca days that we used to get asked on these data. So I think this is probably underestimating the reality a bit. But the survey didn't ask if the readouts were used to predict in vivo or clinical safety outcomes, which we'll come on to in this presentation. So how are we going about this? For each receptor in the Bose 44 consensus panel, we are collating and curating the main or the intended pharmacodynamic effects and the side effects of marketed drugs targeting that receptor for their therapeutic efficacy, as well as candidate drugs that are still in clinical development, but where you've got clinical data or were discontinued in clinical development. So basically any drug targeting that receptor that has both clinical data and KI data at the receptor, or IC50 data. So the sources for the PD effects and the side effects are the drug label, review articles, and also pharmacovigilance databases. So there's FAIRS from the FDA, UDRA Vigilance from the EMA, Vigi Access from World Health Organization, and Canada Vigilance Adverse Reaction Online Database from Health Canada. What we're also doing for the principal pharmacodynamic effects and side effects, we're constructing mechanistic safety pathways. And these are based on the format of adverse outcome pathways which are used in toxicology in the chemical and agrochemical industries. And the value of these is that they ensure the use of a standardized approach for each pharmacodynamic or side effect and each receptor. And I'll show you some examples during the presentation. So knowing the potential side effects from off-target activity of your compound is not enough really. It's very helpful, but what you really want to know is how hard do I have to hit this receptor to see these side effects with my compound? Or put it another way, how much engagement with this target can I get away with before I start seeing these side effects? So do you need to be at the sort of 10% occupancy, 20%, 50% or higher? So this requires knowledge of how much target engagement is required by the marketed drugs targeting this receptor by evaluating the ratio of their effective plasma concentration divided by the Ki or IC50 at this receptor. Then compare the same ratio using the predicted plasma Cmax unbound over Ki for your compound. Does it sit within the range of marketed drugs, or do you have a margin? So if we work through with this fictitious compound Z, so you send off compound Z for screening by one of the screening panel providers. and you get a tabulation of potencies at various receptors in descending order of potency. What you can do, you can plot these out. So you can plot the x-axis is the log of the concentration, or the IC50 or Ki in micromolar. The vertical line is the predicted plasma Cmax unbound of your compound in clinical use, and then you've got the table plotted out for the Ki or IC50 for this set of receptors. So you might intuitively think, well, I need to concentrate on these that are over to the left and maybe ignore the ones that are well off to the right. And what we want to do, and what we're doing, is this. So wouldn't it be good if you could triage these according to the likelihood of interaction with each off-target hit in clinical use? And this would enable you to focus your attention and resources on the secondary pharmacology interactions rated as high and medium. So these would be the ones in red and in yellow here, and hopefully you can pay less attention to the ones in green. So if we look at receptor, one of the off-target receptors, receptor X, and these drugs A, B, C, D, E, and F are marketed drugs that target that receptor for their therapeutic efficacy. And they have a set of PD effects and side effects, some of which are in common. So what we do is roll up the side effects that are in common to all the drugs. In this case, dizziness, headache, and hypotension. And then we can assign a set of these to each receptor as to whether the drug is an agonist in this column or an antagonist. We can also look at what the expected safety pharmacology outcome would be in preclinical species, and also what a useful translatable biomarker might be. And then for each of the side effects, we can draw out these mechanistic safety pathways. So in the same style as the adverse outcome pathway, which starts on the left with a molecular initiating event, we know in this case the molecular initiating event, as they call it, is a receptor interaction. And we're showing this antagonist interaction at this receptor. And we know where the receptor is. We know what the cellular response is, what the response is at the organ level, and then at the level of the whole organism. We keep the references down to a minimum for this so that you can find them from a review article if possible. But the main purpose of this is to provide a snapshot overview of what's underlying that particular side effect. And then we do that for the others that are common to all the drugs. In terms of the quantitative side of it, for each reference drug, we look for all the papers in healthy volunteers on that drug. And in this case, we've got an example of an effect on diastolic blood pressure, which is decreasing over time after a single dose of this drug. What we're interested in is the first data point that's statistically significant. In this case, is this one, and what the plasma concentration was associated with that data point. So here we call that the effective concentration, which is 9 nanograms per mil. We're then using, we know what the molecular weight is, we know what the plasma protein binding is in humans at that concentration level. And so we can convert this to unbound in micromolar units. We know the Ki at the receptor, which we're getting from the IUPAR BPS source references in their receptor nomenclature guide. And then we can do a ratio. the ratio we're doing is the effective plasma concentration unbound over the Ki, or IC50. So where there's a Ki, we'll use that. If the assay provides an IC50, we'll use that. And then we're plotting for each drug the ratio of the effective unbound plasma concentration. So that's effectively this in this example over the Ki for the range of drugs. So the likelihood of compound Z interacting with this off-target receptor X at its clinical Cmax or predicted clinical Cmax. So you're plotting out the reference drugs here, and we've got the range as being considered to be a high likelihood. Arbitrarily, this can be adjusted by the user. Our default is that the medium likelihood extends to 1/10 of that ratio, and then if the ratio is less than 1/10, then it goes into a low likelihood bin. So if your compound falls in the high category, into this range, then it's highly likely that it will be causing these side effects in clinical use. So your options are to look at the side effects and decide whether those are acceptable for your patient population, or you could decide to do further preclinical work to try to clarify whether it's a real issue, or if you like compound rich, if you've got other compounds, or if there are other issues with this compound, you might just drop it. And it really depends on your risk appetite and the strategy around the project that the compound's from. If instead it falls in the medium category, again, you may decide to follow it up with in vitro and in vivo work to clarify what its risk is from your animal studies and in vitro studies. Whereas if it falls in the low category, you probably don't need to do any more investigative work on it. You probably want to keep an eye on it because often the predicted pre-Cmax creeps up, so this will move this more towards the medium, and if it's already at the border, it might start to encroach on it. But it gives you a triage of the risk of the compound interacting with this off-target receptor, which is a handy guide to how much extra work you need to do on it. So again, this is similar to the previous plot of assays, but move through 90 degrees. So we're going from left to right on here, from in vitro receptor binding, in vitro receptor functional assays, in vitro complex functions, in vivo, and then through the three clinical phases and onto the market. And various in silico models within the development that we're undertaking in ToxStudio will interact with these various steps. But behind that, this diagram shows 4 routes of forward translation from in vitro data. So route 1 is really going straight from receptor binding to patients or to the market, which is quite a leap. Route 2 is what I've just been talking about, going from the receptor assays through to the phase 1 predictions. We feel this is achievable. Route 3, you're taking into account some of the more sophisticated in vitro outputs and also the in vivo studies. And then route 4 is at the early in vitro stage. So what we're doing with secondary intelligence is, in this diagram, is route 2, which is going from the in vitro assays through to predicting phase 1 with input from in vitro and in vivo at various points. Now, we will need to apply PBPK modeling to account for, these are two examples, there are other examples as well, but for example, receptors located in the CNS. It wouldn't be appropriate to use plasma concentrations for adverse effects or side effects that originate in the CNS that require penetration of the blood-brain barrier. So we'd have to adjust the concentrations of the test compound using PBPK modeling. Similarly, for receptors located intracellularly or for receptors that the drug has to get into the through the cytoplasmic membrane to access its binding sites that may be located in the membrane, and also for readouts from functional in vitro assays, then we've got the SIVA toolkit to apply there. So just to go through some aspects of the secondary intelligence software. So we've got the receptors in a panel view, and this is the Bose 2012 consensus panel of 44 receptors laid out in a sort of periodic table format with ion channels, GPCRs, enzymes, kinases, nuclear hormone receptors, and transporters. So your test compound would light up this panel from its readout. So the receptors that have no interactions are grayed out, and then the ones that do have interactions are given the rating of high, medium, or low likelihood of interaction, and the type of interaction is indicated by this symbol, either antagonist or agonist effects. And then you would go into each receptor where you've had an interaction, and you saw this upper panel on the left before, which will take you to what the highlights of the side effects are. A list of other drugs that target, or marketed drugs that target this receptor, with links out to Campbell and the IUFAR database, etc. In the top right panel, you've got the input for the predicted free Cmax for the compound. And then this plot in the bottom right is what I showed earlier schematically, which has plotted the various marketed drugs that target this receptor, and then where your compound sits on this spectrum from green, yellow, and amber, sorry, green, amber, and red. In this case, the compounds come out within this cluster of marketed drugs and is rated as high likelihood of interacting with that receptor. And you can visualize the compound across a panel of receptors. So they're organized here, going from red down to green. And you can also view multiple compounds shown here and compare them. And also for one of the compounds, you can see how far into the green they are, how far into the red they are, etc. So multiple ways of visualizing, which is helpful to get your head around the readout from the screening panel. The software will also auto-generate a report if required, and this could be appended to a report of a screening panel and form part of regulatory submission. So you wouldn't have to enter much data to generate these. It would just be like an entry panel. So in summary, we've brought together safety pharmacologists and mathematical modelers to address the needs of safety pharmacologists. We want to work closely with you to tailor ToxStudio to your needs. Secondary intelligence is the first tool in the ToxStudio toolbox and will address off-target hits in your screening panels. And there are 3 ways for you to access secondary intelligence. So the first is to join our QSTS consortium. And if you do, then additional receptors are included as and when they become available, plus other in silico tools that are ready now. Also, more importantly, further QSTS tools will be developed in ToxStudio, guided by consortium member priorities. The second way to access secondary intelligence is to purchase an annual software license. And the third way is via consultancy projects. So we would run your compounds through the tool and provide you with a report. Just like to thank my colleagues in the team and elsewhere in Satara, and Lynn Rosenbreyer-Ribeiro, who's helped us with the science side of this tool. And very happy to take questions and answer them. Thank you for your attention.
All right, thank you so much, Will. As mentioned, we'd be happy for you to put your questions into the Q&A panel for Will. Looks like we have our first question from the audience. Someone says, the target engagement methodology will likely be easier for peripheral targets. It is also likely easier for antagonist inhibitors rather than, is it also likely or easier for agonists or inhibitors rather than agonists? Is the spectrum of ease from a peripheral antagonist to a central antagonist?
Could you just repeat that last bit again, Suzanne? Last sentence. Is it likely?
So they were wondering what the spectrum of ease is from a peripheral antagonist to a central agonist.
Oh, I see. Yeah. It's a good point about agonists, because the properties of basically binding affinity and intrinsic efficacy are two different properties of an agonist. So we're focusing on the binding in terms of the Ki for now, and using the ratio between the plasma concentration and that binding. The reason being for the radioligand binding Ki is that that's a much cleaner, reproducible measure that is less assay dependent than in the functional assays for agonist efficacy. But it is a good point, because it makes it more complicated for agonists than for antagonists. So yes, I put antagonists at the if you like, the easier end of the spectrum. And then the other part of the question was for peripheral versus CNS. So for peripheral actions where the plasma concentration is, to all intents and purposes, in equilibrium and is about the same as the concentration at the receptor, that's relatively straightforward. I think two areas where there's more of an issue is for CNS. So obviously you've got to factor in the free brain concentration of your compound, which you can predict to some extent. And another area is in the gastrointestinal tract. So given that most drugs are administered by oral route, you, particularly in the upper GI tract, you're going to hit them with a higher concentration than is necessarily your plasma Cmax. So that's a challenge. But the point I would make is that we have these challenges already. We already have to deal with that. And this isn't, if you like, a magic solution to deal with it. We're going to have to surmount those challenges as well. But at least we can do it in a consistent way and ensure that we do it reproducibly each time. So we will become, or the tool will become more sophisticated as we go along. But yeah, I think the spectrum would be peripheral antagonist and central agonist would be the both ends of the spectrum.
Someone says, if we assume equivalent engagement necessary for efficacy and off-target safety effect, you'd need the intrinsic selectivity of 10-fold to be medium risk and 100-fold to be low. So they want to know if that's correct.
So if the safety I see. So if the safety, if the side effect was occurring at the same concentration required for the primary activity, then you're right. Then we've got like a tenfold shift down to get to the low, to get to a low likelihood of interaction with that off-target receptor. Yes, that's right. But if you've got the side effect of that, or the potency of that receptor is equipotent with your primary target, you're going to struggle to move away from it. You're going to have to design it out of your molecule, unless the side effect is considered acceptable for your patient population.
Someone says, this is a very interesting presentation. Could you talk about some of the differences or benefits between ToxStudio and other software?
I can't really compare and contrast with, if you like, software from our competitors. But I think in terms of what we're trying to do, it's in safety pharmacology, in Silico, we're probably in uncharted waters in that we are forging ahead with this, where there aren't any other options at the moment. Whether there will be in the future, that's another question, but certainly at the moment, I think we're pioneering this approach for safety pharmacology.
Someone says that they have two questions, so I'll ask them one at a time. Do you have to have Ki IC50 data for secondary intelligence functionalities, or could it work with the percentage of inhibition?
Yeah, that's a good question. And different companies have different strategies for their secondary pharmacology, where some may just run the compounds, say at 10 micromolar or 30 micromolar, and then just use the percent inhibition and not trigger a full concentration effect assay. So we might be able to do an extrapolation to predict the Ki using an assumption that the hill slope is 1 from the percent inhibition. It wouldn't be ideal, but if that's the strategy the company's using, we can do some predicted KIs from that data.
The second part of the question was, are you also planning to integrate QSAR models to predict off-target activities, i.e. IC50s for compounds based on their structure only?
Yeah, that's a very good question. Not in version 1, and possibly not for a while, but QSAR models do exist for numerous receptors. I think probably the least we could do is at least have a link to either the model or publication on the QSAR model for that receptor, if that would help the chemists, although they probably know what they are anyway. But I think going forward, it would be nice to be able to do that. And then the chemists in particular could, if you like, play with the secondary intelligence tool by looking at different, using QSAR and getting, if you like, virtual readouts of what their molecule would do in terms of its off-target effects. I think that would be something for the future, but it's certainly on our radar.
Great. Someone wants to know if you can summarize the workflow of ToxStudio, in particular, the balance between information collected from the literature and compound-specific information from a current drug in development for which one wants to have a safety prediction.
I'm not entirely sure what the question is getting at, but what we're currently doing is there's two aspects. Well, there's two sides to this equation. One is the Ki, which is we're going to sources via our UFAR BPS, which has quite a good set of references to go at for the source data. And then on the clinical side, we're looking at clinical pharmacology publications in healthy volunteers. And the reason for that is that the plasma concentration you need for therapeutic efficacy will vary between indications. So some drugs are often repurposed or used now for several indications, and you will find that the plasma concentration you need varies between the indications. So if you were to just look at the therapeutic plasma concentrations, you may be well above what you would see with your compound in as an off-target effect. So you might have to hit the receptor harder to get a therapeutic effect, if you like. In terms of workflow, we're looking at those sources, so the KI sources and the clinical pharmacology sources. Then for the adverse effects in the mechanistic safety pathways, again, these are via the literature to basically back up the key side effects of that class of drug, or what happens when you're an agonist or an antagonist to that receptor. So it's mainly a literature-based project.
Looks to be one final question. I noticed in one of the secondary pharmacology visualizations that various receptors off targets have different ranges of exposure coverage for low, medium, and high risk. What is the basis for what constitutes the different ranges for each secondary pharmacology target?
I'm not sure what the question is. There's a problem with typing in questions, unfortunately, but perhaps they could email me afterwards and I can try and clarify that. If you meant there's a range for a set of reference drugs, there can be a range of ratios, which is to be expected, and it's difficult to explain in terms of why there is a range, why they're not all sitting on top of each other. But no, I didn't quite get the question, so perhaps they could email me afterwards. I'm happy to address questions by email.
Okay, great. Thank you so much, Will, and thank you so much to our audience for all those excellent questions. Will, if you can advance to the next slide, please. Just a few announcements before we leave. Thanks everybody for participating in this webinar. On behalf of Certara, I would like to thank you for attending today's presentation. This concludes the webinar. Goodbye and have a great day.